Okay, so last time we essentially finished up this long story of catabolism that we had started several weeks ago, and we've talked about anaerobic respiration, so alternative terminal electron acceptors, and then went through this whole set of where other sources of where the electrons can come from. So a couple of review questions from last time. So the first question. Sorry, I heard somebody say something, but I did not hear the answer. D? I heard D, I heard B. A. I hear A. So now I'm hearing 3 answers. Okay, so all right, so this is for bacteriorhodopsin. This is like the first system that we talked about. So this one is actually retinol. Yeah. So the chlorophyll and the antenna complex, they're related, right? So chlorophyll's in the antenna complex, and those are for the photosystem type systems that we talked about. All right. So how about the second question? All right, so I only heard one answer, which is B, which is correct. So what does this actually mean, though? What is the cyclic nature of this? Like,
it pumps it back into, or it pumps the electrons back into the electron transport system. Sorry, not the electron transport system. The first, you know what I'm talking about.
I do know what you're talking about. No, yes, thank you for sharing. So it goes through the ETS, but then it goes back. It'll send the electron back into the original bacterial chlorophyll, and then that can get re-excited again. But yes, thank you. Okay, so today we're gonna do the reverse, and so we spent a lot of time talking about catabolism, but we're essentially only gonna do anabolism for one day. And you'll see when we get there, I don't expect you to know a lot of the things, but we'll just kind of focus on the main ideas. So why don't we just get started? So biosynthesis or anabolism is building of complex biomolecules. So again, it's the reverse of catabolism. So essentially, for all organisms, they require all the essential elements. So examples: carbon, oxygen, hydrogen, nitrogen. And then if you think about what other kinds of molecules that the cell needs to build, to grow, and for it to function, you can think about what other essential elements might be necessary. So they also require reduction. So biosynthesis is usually reduces the substrate by either hydrogenation and then by removing oxygen. So this is usually done by reducing agents such as NADPH. Remember we talked about, we've been generating NADPH in some of these reactions, so we're going to start using them now. Again, these all require energy. And so building, as you can imagine, building complex structures requires energy, and the reactions tend to be coupled with the hydrolysis of ATP or oxidation of NADPH. Sometimes can be coupled to fluxes of ions down transmembrane gradients. So here's just an overview of an example of what we mean by biosynthesis. And so this picture should the shapes of these pictures should kind of look familiar to you, right? These are the processes that we had talked about, but now all the arrows are going in the opposite directions. And so this looks like the TCA cycle. So this is glycolysis. And you can see now that we're trying to build up to all these different things. Even the initial catabolic reactions that we had talked about, breaking down some of these things, now we're trying to again go back and build these up. So we'll talk about some of the different cycles to do this. But so first you actually need the materials to do it, right? So where do the cells get the materials for biosynthesis? They can come from metabolic intermediates from glucose catabolism or the TCA cycle. And so I guess in general, when we were talking about the theoretical maximum yields of ATP when things go through glycolysis and things like that. We are always theoretical, and it's always the maximum yield, because at any point, if the cell needed one of the intermediates to make something else, that would just get taken away at that point, right? So this is where some of that imbalance happens. So you can use these intermediates from these cycles themselves, or there are actually some autotrophs that can synthesize entire sugar molecules starting with CO2 and just going through the whole process in reverse. But as you can imagine, if a cell were to do that, that would again be very expensive, but not only in terms of energy, but also in terms of just maintaining all those pathways and producing all the enzymes and getting all those genes made to make all of those steps. And so oftentimes what the cells will try to do is to get them from the environment or get them from other cells that are making them, whatever intermediate they can. And so they will either lose or downregulate a pathway if the product is available from the environment, because it's going to cost way more for them to have to turn on the genes, make those proteins to be able to make all these components than if they can just steal it from somebody else, if it's already given to them. And so examples of lost pathways, so in humans, we've lost the pathway for making vitamin C because we get it elsewhere, and then also just in the case of parasites, so parasites lose many pathways because they are essentially living off of their host, right? All right, so here is again one section of kind of that big overview that I showed a couple slides ago, but just to kind of point out where some of these intermediates might be going off to. So just some examples. So pyruvate, again, so this actually can lead to many different things, right? And some of the other, so even some of the smaller like G3P can go into lipid formation. Coming down here in the TCA cycle, you have succinyl-CoA that's can be branched off and going into other amino acids, or even some vitamin B12 synthesis here. So again, this is just an overview to show you where some of these intermediates can be taken in terms of going back in the other direction and doing biosynthesis. All right, so the main one that we're going to go through is talking about CO2 fixation in the Calvin cycle. So you've probably heard of the Calvin cycle before in your other classes, but the Calvin cycle is also known as the reductive pentose phosphate cycle. So this is the primary source of Earth's fixed carbon. It's conducted by several categories of bacteria, including photoautotrophs, being the cyanobacteria, and then chloroplasts of algae and plants. There are facultative anaerobic purple bacteria, and then some lithoautotrophic bacteria that use energy from metal oxidation to do the carbon fixation. So it's not every bacteria can do this, but this is where the main source comes from, these bacteria. All right, so the Calvin cycle can be broken down into three main phases. So the first phase is this carboxylation and splitting. So going from first making the 6C molecule and then splitting that into 2 3-carbon molecules. So this is done by the enzyme Rubisco, which we'll talk about in a few slides. But Rubisco essentially mediates the condensation of ribulose-1,5 bisphosphate with CO2 and water. So this is where the 1 carbon comes in, that the CO2 that it's fixing. And that leads to, so this is, the ribulose is a 5-carbon molecule, right? So now this is 6, but this is actually an unstable intermediate, so it immediately hydrolyzes into 2 3-phosphoglycerate, so 2 PGA molecules. And so one of these molecules will contain the carbon from the CO2. The second phase, so now we're reducing each of the PGA molecules to G3P molecules. The thing to remember from the second phase is that it's requiring ATP, and then also it requires NADPH to do this. So again, we're using up all this energy to make these molecules. In the third phase, so now in this cycle, a key part of this cycle is actually regenerating this ribulose 1,5-bisphosphate so that it can get reused again. So now then it's like, well, if I'm just gonna reuse, so then what happened? Like, how am I actually gonna start making molecules for using some of that for biosynthesis? One of every 6 G3P molecules, so will actually can exit the cycle and be used for biosynthesis. So the other 5, those are the other 5 that they go through a complex series of reactions, and I don't expect you to know those reactions, but those are going to get used to regenerate the ribulose 1,5-bisphosphate so that the cycle can go through again. Oh, so these complex reactions that that convert, so you ended up with G3P, the reactions that you need to go back to ribulose 1,5-bisphosphate. Okay. Yeah, you don't need to know those. Okay, so just like with the other cycles, I'll kind of just write down some of the key steps and kind of track the carbons. So this one is relatively easy for tracking carbons compared to the others. Oh, sorry, yep.
Sorry, I have a question.
Yep.
You said there's a previous slide?
Yep.
What reactions?
All right, so. Okay, so again, we're gonna start with the ribulose 1,5-bisphosphate. And this one has 5 carbons. So this is going to, so this is where the CO2 comes in. So remember, this is CO2 fixation that we're talking about. So the CO2 comes in here. This is done by Rubisco. And this is now an unstable 6 carbon, so this automatically just goes to 3-phosphoglycerate. So this is AGA. So now this we had 1 carbon coming from this CO2, and so we'll end up with 2 of these, and one of them is going to have the carbon from the CO2. So now what we want to do is go from here to glyceraldehyde 3-phosphate. So this is G3P. So here, the key for this here is that it's going to take ATP and NADPH. And this is per PGA. You still end up with the 2 3-carbon molecules going from here to here. You still end up with the 2 3-carbon molecules. So then you're going to end up, oops, wrong color. You're going to end up, so these are the reactions that you don't really have to know, except for that they happen. So this gets you to ribulose 5-phosphate, which at this point now it's back to 5 carbons. So whether or not it's the one from the CO2, it's hard to keep track of at this point, but so the other thing here is that you're going to use another ATP to get to the from the just the 1-phosphate to the bisphosphate. You're going to use another another ATP here. And so so one of these can go off to biosynthesis, but this only happens 1 out of 6 G3P.
Can we go over how we look from NADPH we went to NADH? Is that interesting?
So this is just standard. I mean, for it's just gonna give the electrons back. It's kind of it's more than you need to know for how the actual reaction for the rearrangement for this. Just know that it uses that energy.
So the phosphate leaves the NADPH and just becomes.
Oh, sorry. Now I see why I did not write this correctly. Yeah, so this should be an ADP. Sorry, that's why. Is that what your question was? Sorry, I didn't realize. That was a mistake. It should be an ADP.
What is the 1, 6 G?
So 1 out of 6. So for this biosynthesis, so every 1 out of 6 can exit the cycle and used for biosynthesis.
So if it happens 3 times because there's 2 of them, makes it
Yep,
okay.
exactly.
What makes it 1 out of 6 times?
I don't know. So it's going to come from the fact that it needs to regenerate the 5 carbons, and they can't just like all go away. But I don't know why it turns out that they were just 1 out of. So every 3 turns, like I don't know why that was the case.
Would you want us to know the key steps? Would you want us to know that Rubisco makes it 3?
Yes, and we're going to talk about Rubisco in the next couple slides. Yep.
So what's the total net yield for?
The total net yield, so that's a little bit complicated depending on what you want. So I would say that
Do we have to know it? The net yield for this?
so just think about it in the terms of like, this was drawn for 1 CO2, right? But in reality, you need it to go through 3 more times for you to actually be able to get one of these to go out. And so you can kind of track things that way. But so imagine if it was, so now from here to here, you actually use 2 ATP, right? And 2 NADPH. But that was for 1 CO2. So you just multiply that by 3 for it to go through for you to end up with 6, the 6 down here.
Okay. Can you say 3 more times? So it needs to go 4 times or 3 times?
3 times.
3 times.
3 total, yeah. Any other questions about this?
Professor,
Yep.
where do the carbons come from to make the ribulose 5-phosphate?
So do you remember the pentose phosphate pathway?
Oh, okay.
Yeah.
All right.
Any other questions? Okay. So now we'll talk about Rubisco a little bit. So Rubisco, again, this is the key enzyme that you should remember that's associated with the Calvin cycle. It is also actually the most abundant protein on Earth. So this is unique to bacteria and chloroplasts. It is made up of several small subunits and large subunits. The large subunit is actually where the active site is contained. The function of these small subunits is not super clear. But so while the general protein is conserved, the actual number of how many S subunits and L subunits there are varies from species to species. And so again, just as a reminder, so this is the reaction that it's catalyzing. So the ribulose 1,5 is phosphate, and taking that with the CO2, and then it makes this unstable 6-carbon intermediate, and then you end up with the 2 PGA molecules. All right, so a little bit about the regulation of the actual Calvin cycle. So there's an enzyme system called the carbon concentrating mechanism. So this converts CO2 to bicarbonate. So this is key because this is how the cell actually keeps CO2 in the cell. So CO2 can diffuse freely across the membrane. So if the cell wanted to hold on to some CO2 to be able to do these biosynthesis reactions, it actually has to be able to hold on to it somehow, and so it converts it to bicarbonate to be able to do that. So many organisms contain the rubisco complex in what are called carboxysomes. So these are these polyhedral structures here. So some of these structures in here. So these are found in CO2 fixing lithotrophs, cyanobacteria, and chloroplasts. So the carboxysome is what takes up the bicarbonate, and then it's converted back to CO2 by carbonic anhydrase. So the rubisco is still using CO2 regardless of the fact that the cell is trying to hoard CO2 by changing it to bicarbonate. It still has to be converted back. And so it's using carbonic anhydrase to do that. So then rubisco can go on and do its thing. All right, so that's kind of the main CO2 fixation pathway, but we'll go through a few alternative ones. So we have the reductive or the reverse TCA cycle. So most of the individual reaction steps of the TCA cycle are reversible. Meaning they have small delta Gs. So there are steps that release CO2 in the forward direction. So if you go back and review, you'll notice that every once in a while the CO2 is coming off. And those may assimilate small amounts of CO2 in the reverse direction. So when it's going in the reverse direction, those reactions have to go in reverse, right? Also, so some of that CO2 can be taken back in that way. In general, all organisms fix small amounts of CO2 through special reactions that regenerate TCA cycle intermediates. So those regeneration steps are called anaplerotic reactions. So one of the examples is here. Is this reaction here? So this is taking PP and CO2 and getting back to oxaloacetate. So that's just an example of one of these anaplerotic reactions. So still a little bit more about the reverse TCA cycle. Some anaerobic bacteria and archaea can run the entire TCA in reverse and generate acetyl-CoA. So that this uses 4 to 5 ATP to fix 4 molecules of CO2 and generates 1 oxaloacetate as the kind of backwards product of this cycle. So again, we talked about this mostly in the context of glycolysis, but the enzymes in the TCA cycle, again, a lot of them are the same because they're reversible, except for the ones that are these ones that are highlighted in yellow. So these are the ones that are spending ATP, and those are the ones that are unique for the reverse cycle versus the forward cycle. So the reverse TCA cycle feeds the reverse glycolysis, which is known as gluconeogenesis. And like I said, some organisms actually will from scratch make these molecules just by fixing CO2 through these pathways. The reduction in these cases can be formed by NADPH or NADH, depending on the reaction, and then also some reactions can use this reduced ferredoxin instead. Yep.
In all these cases, we're making either glucose or some other carbon source?
Yeah, so it's really going to depend on what the cell needs. So if you look at that big picture overview, it's like, what does the cell need at that time? If it can, if the cell has the ability to run these pathways, then it'll make whatever it needs at whatever time. Okay, so another alternative pathway is this reductive acetyl-CoA pathway. So in this case, you get two CO2 molecules, and they're condensed through converging pathways to form the acetyl group of the acetyl-CoA. So this is used by anaerobic soil bacteria and methanogens. In this case, the reducing agent is H2 and not NADPH. So this one, so what it means by converging pathways, so here the actual two CO2 molecules, they actually go through two different reactions. And so the first pathway here, so this goes through this formic acid intermediate, and then I don't know why they changed it from highlighting it in green to yellow all of a sudden, but here's the one, so if you're tracking this carbon dioxide, the carbon is here. And then this one will come together with, so the second CO2 actually goes through a carbon monoxide intermediate. And then so these two come together with the CoA to, in the end, give you acetyl-CoA. One more alternative pathway is this 3-hydroxypropionate cycle. So these are used by Chloroflexus bacteria and some archaea. So in this case, CO2 is fixed by acetyl-CoA into 3-hydroxypropionate. And so this is reduced by NADPH. In this case, you need 3 molecules of CO2 to make 1 molecule of pyruvate. So this is a way for the cells to get back pyruvate. All right, so just one question for discussion. So we've been talking about running some of these cycles in reverse, right? The TCA cycle and glycolysis. So why do bacteria use some enzymes in both directions, whereas other steps require different enzymes for the catabolic versus the anabolic direction? So not only like why do they, or why can they actually use these enzymes in both directions? So think about it for a minute, and then you can share your answers. You can discuss with your neighbors. All right, any ideas? Anybody want to share their ideas of why this might be the case? I could tell that you guys are discussing it. I know you were discussing it, so somebody share something. I'm not going to tell you the answer unless somebody gives an answer. Yeah.
It has something to do with the carbons, maybe?
What about him?
I have no idea. I'm just saying.
Okay, so just throw something out there. What might it be about the carbons?
So, like, for example, the TCA cycle, the enzymes are used to release carbon in the form of CO2. So, I guess enzymes that can do that can take in CO2, can be used in both directions.
Okay, so maybe the components that the enzymes are able to recognize,
Yeah.
right? Okay, Rachel, you had something.
I was thinking something about the environment, whether or not it can deal with those expenses.
What do you mean, deal with those expenses?
I have no idea. I just guess.
Something about the environment, like meaning the environment that the cells are actually in?
Yeah.
And whether or not, what about it? No.
No idea.
Okay. Okay. Anybody, something new, or also maybe expand on what Rachel's idea. Go ahead, you guys.
I have something else.
Yeah, that's fine.
So, did we say that the enzymes that are switched have involved like ATP, like making ATP, or those are the ones in
The ones that are
TCA cycle,
switched?
we said, or did you say that they're all the same except for the ones that make ATP?
That require ATP. Yep.
Or that requires, so usually when something's phosphorylated, there's a conformational change. So
Okay.
now an enzyme has to recognize a different structure.
Okay.
And then there's just more of a change compared to other things.
Yep. Yep. You had something.
I was going to say it has to do with like their activation energy and
Yep.
the delta G.
Yes. So what about the delta G about a reversible reaction? What is it about that?
Different, I guess, or lower or higher, so that we need a different enzyme.
In which case?
In the reverse.
Yes. Yep. So something.
Energy will be lower, so we would need a different enzyme.
Okay. So let's talk about this a little bit. I know you have something. I'll come back to you in a second. So let's talk about this. Make sure that everybody understands this concept first. So in the cases of these reactions, there are delta G's, and I mentioned this. So these reversible reactions, they have small delta G values, right? So we talked about this a few lectures ago. What can influence delta G? Sorry. Enzymes. So that can actually, so that's going to make the reaction faster. That's not going to influence the delta G. Temperature, pressure, whether reactants. Sorry, what did you say? Yep.
Delta S.
Yep. So in the case of these reversible reactions, somebody over here said reactants. So the concentrations of the products and the reactants and the products are going to play a huge role, right? So if one end is around, if it's going to shift, it can shift the delta. Remember that the concentrations can shift the delta G negative or positive. And so depending on the situation in the cell, so these reactions, if the delta G's are very small, but all of a sudden there's no reactants in this environment, in this state of the cell, then the reaction's going to go from product to reactants, right? It's going to go in the reverse direction. Okay, somebody else had something and I forgot who. So there was it you? You were going to say something else, something completely different, maybe? It's okay if you weren't, but.
I was just going to say, like, some of the kinases and phosphorylases, like one is adding phosphates, so wouldn't that be
Yep, so that's similar to what Aaron was talking about with the ATP. Yep, exactly. Anything else? Yeah, so the only thing I would want to add is for you to just think about what the enzyme's actually doing, right? So the enzymes they're not actually doing a reaction per se, right? They're just bringing the reactants together. And so think about what the reaction's actually doing. And for reversible ones, all they have to do is bring the two things together. And so if it's in the same place, and this is where we're talking about concentrations dependent, then it can easily, that same enzyme can just be used again to do the forward versus the reverse reaction, because they already are made to be able to fit these two different chemical components, right? In the sense of your irreversible reactions, so like Aaron said, with the things that are, and also, what is your name?
Bendros.
Bendros. Okay, so what Bendros and Aaron were talking about where you were dealing with phosphorylation and dephosphorylation and creating a product that now the enzyme might not actually recognize, right? But the also key thing to remember is that it's going to use the ATP, and so making ATP from ADP is a completely different reaction that the enzymes might not be able to accommodate. All right, any other questions? That's good. Yep.
So what's like the solid answer?
So I mean, everybody gave good answers. I think the what I just talked about remembering what the enzyme is actually capable of doing, but also in the reversible reactions, the delta G's are small and they're very much dependent on the reactants and the product concentrations, and then just for the irreversible reactions, the ones that are requiring ATP, that's not something that you can just use the same enzyme to go back and forth with. And then also just as a side thing, those ones that are requiring ATP, those are the ones that are going to get regulated depending on the state of the cell. So if the cell is in need of making something, or if it's in need of energy, it's going to turn on making those enzymes. And the cell doesn't necessarily want to be just having to turn on special enzymes for every single one, because that's going to cost them a lot of energy, right? So, okay. So we'll talk a little bit about the biosynthesis of a few of the other key molecules. So the first one we'll talk about is fatty acids. So fatty acids are built from repeating units. So this is done by the fatty acid synthase complex. It contains all the enzymes and component building proteins bound together in proximity. So it's just kind of a conglomerate of proteins that are going to do this reactions. And the bacterial version is FAS2. So in this case, so now again, we're going to be using acetyl-CoA, right? So this is one of your intermediates. We're going to use this, and so this is if the cell needs to start making fatty acids, it's going to get tagged for fatty acid biosynthesis by adding CO2, which makes it now malonyl-CoA. And then the CoA is actually replaced by this acyl carrier protein, so this ACP, making it malonyl ACP. So this is kind of the start of fatty acid biosynthesis, is with this process ending in malonyl ACP. So at that point, units of aceto-ACP, so again, it can come from acetyl-CoA with just changing it to this ACP. So these are then added repeatedly, and they're reduced twice by NADPH. So this increases the length by 2 carbons because the acetyl group has 2 carbons, right? So most of your lipid chains, they're even carbons because they're produced this way by 2 carbon molecules, by the addition of this repeating of 2 carbon molecules. And so depending on the cell or how long it needs, it's just going to keep just doing this cycle until it hits the length that it needs. And these can get added to glycerol or NAG to form lipids, or proteins to form lipoproteins. So again, just depending on what the cell needs at that point, it's going to decide what's going to get added to where. All right, so the other key thing is so we've talked about carbon dioxide and getting carbons, but the other key thing is where do we get the nitrogen from? So in macromolecules in the cell, so they're virtually all of the nitrogen is reduced. So the bacteria and archaea, they must reduce the nitrogen completely to form ammonia, which is mostly protonated to ammonium at pH 7, so whatever the state of the cell is at. So while nitrogen is the ultimate source and sink of biospheric nitrogen, several oxidized or reduced forms are found in the environment and produced by living organisms. So we talked about this several times before about how different bacteria, just different bacteria in the earth, they can fix nitrogen, but not all of them can actually fix the N2 gas, right? So it's very specific what bacteria has the enzymes to do which ones, and so whether it can take the nitrogen gas or whether it's taking nitrite or nitrate, that's going to depend on the bacteria and the enzymes that it has. But again, so we're going to just talk about what happens in the form of the nitrogen gas. So the mechanism of nitrogen fixation is hugely energy intensive. And for the species that can do this, it's largely conserved. So here we have N2 going to ammonia, but you can see we got also, besides just 16 ATP, we also have electrons, right? So that also is a form of energy to the cell. So electrons are donated by NADH, H2, or pyruvate, or sometimes they can be obtained through photosynthesis. So it's about 3 ATP equivalents per 2 electrons, so a total of about 28 ATP per nitrogen fix. So this is a lot of energy for these cells that can do this. The overall conversion is catalyzed in 4 cycles by the enzyme nitrogenase, which we'll show a picture of on the next slide. Nitrogenase has 2 different subunits. One is the Fe protein that contains an iron sulfur core, and then it's the Fe-Mo protein that contains iron and molybdenum. So here is what this looks like. So here's this process here that goes like this. And so the electrons, they're acquired by the Fe protein, so this requires energy from ATP, and then they're transferred to the Fe-Mo protein to reduce nitrogen. So they're acquired here and then transferred here to this Fe-Mo protein. So the Fe-Mo protein binds to H+, so binds those here, which are reduced to hydrogen gas. So at this point here, the nitrogen gas can bind to this active site and it displaces this gas. So the N2 is coming in here, which is displacing the hydrogen gas there. And then it goes through several steps where the electrons are used to reduce the substrate. So it goes from N2, so now you see now there's the 2 H's here. And then, so this is just kind of it goes from here to here. And then again, more electrons required, and so now you get more hydrogens on them. And then again, now you end up with the 2 ammonia, which in the cell it will generally end up protonated to the ammonium form. So again, this is successive pairs of the H+ coming in here to reduce the electrons and the H+ to reduce this all the way to ammonia. Just that it's you have to do it in steps, right? So that one that is this nitrogenase enzyme that is doing this whole process, but it's going to take lots of ATP and lots of electrons to do this, and so it's going to take in the nitrogen and it's going to do it 2 electrons at a time. It's going to get reduced 2 electrons at a time until it ends up at ammonia. Let's see. All right, so cells can actually only do this in an anaerobic environment because of all the reducing power. If oxygen's around, that's just going to kind of mess up the situation. It's actually not going to be able to put the reducing power towards the nitrogen instead. And so so organisms that are aerobic, they've developed several solutions to this. Some of them have protective proteins that stabilize nitrogenase and prevent attack by oxygen. So here's where the correction was for the version 2 notes. I was missing a couple of words here. So the rhizobia, the plant hosts, they produce leg hemoglobin, which actually sequesters oxygen away from the bacteria so that they can actually do this nitrogen fixation. Some cyanobacteria fix N2 only at night when they do not conduct photosynthesis, so they're not releasing oxygen. Remember we talked about the oxygenic Z pathway last time, and they're making oxygen, so they can, but they need all the photons to be able to do that, right? So they'll switch and only do nitrogen fixation at night when they're not actually making oxygen. And then some have these specialized cells called heterocysts that are specialized for only doing nitrogen fixation. All right, so now we'll talk about amino acids and nitrogenous bases. So how do the cells make these? When possible, they're going to try and get them from the environment. They don't want to use the energy to do this, so they're going to take it from the environment if they can, but depending on where they're living, competition for these nutrients can be really high. And so most of these free-living microbes and plants, they have the ability to make all the standard amino acids and bases, as well as the non-standard variants and the ones used for cell wall and tRNAs. So some are made from relatively simple biosynthetic pathways, others are much more complex, so we're not going to cover all of these pathways. I will just show you kind of the key thing that I want you to know, but just also to just realize where some of these components might be coming from. So here, so we have the carbon skeletons for the amino acids. They can arise from diverse intermediates from metabolism. So again, taking these different metabolic intermediates, they can be used to make some of these different amino acids. And then one way to kind of conserve energy is through these branched pathways, which early intermediates are used to form several products. So like for instance, for aspartate, so if you go from oxaloacetate to aspartate, this can then be used to make several other ones. And so that's a way for the cell to kind of conserve resources and not have to make a ton of different enzymes if it uses some of these pathways like that. All right, so the one thing that I do want you to know is how does it actually, so say I got the ammonia from some cell that was able to fix the nitrogen gas, right? So how does it actually put that into an amino acid? So ammonium condenses with 2-oxoglutarate to form either glutamate, or it can go with ammonium with glutamate to form glutamine. So this requires reduction by NADPH in the case of glutamate, or ATP in the case of glutamine. So this here, so here's the first reaction. So going from 2-oxoglutarate to glutamate. So this again is where the ammonium is coming in. This is done by this glutamate dehydrogenase enzyme. And so this is how you get the ammonia into the glutamate for the amino acid. And then the other case is where you can put in another one. So you can do this and it can get converted to glutamine. So this is an example of this transamination, so transfer of ammonia between two metabolates. So the ammonia gets transferred from here to here. All right. That is basically it. And just a reminder that your exam is a week from today. And so it's going to be a similar format to last time, so just kind of refer to all of the intended learning outcomes for what you want. Probably not, because of the observation. They need a little bit of time. They're going to want to talk to you guys too, so yeah.